Welcome to Microfluidic-enabled manufacture of polymeric nanoparticles, from size tuning to seamless scale-up. I'm Troy Pereira, Marketing Communications Associate at P&I. Before we begin, I'd like to remind our audience that our second annual Nanomedicine Symposium, entitled Nanomedicines, Enabling New Therapeutic Modalities, takes place on July 15th in Cambridge, Massachusetts. We'll be live streaming this event on both our YouTube and Twitter pages. To learn more and to register, please visit pnicambridgelivestream.eventbrite.ca. It is my pleasure to introduce today's speaker, Dr. Shyam Garg. Dr. Garg has over 8 years of experience in the development of nanomedicines with a strong focus on polymer delivery systems. After completing his pharmacy degree from the University of Mumbai, India, he earned his PhD in pharmaceutical sciences at the University of Alberta. He is currently using P&I's technology to develop new applications in polymer nanomedicines. Now, some basic housekeeping. If you have technical issues, please use the question or chat box and we will do our best to troubleshoot. If you have any questions for our speakers, please use those same features and we will try to answer as many of them as possible at the end of the session. If we do not get to your question, we will answer them after the event via email. Now, without any further delay, Dr. Garg, take it away.
Thanks, Troy, for the introduction. So today we're going to talk about how we can make polymeric nanoparticles using microfluidics, and we'll also discuss about controlling size as well as scaling up your nanoparticle formulations. So today, polymeric nanoparticles are being researched extensively as drug delivery vehicles due to a number of factors which include their chemical flexibility, controlled release properties, good biocompatibility and biodegradability, as well as their ability to be used as multifunctional carriers. Various types of polymeric nanoparticles can be synthesized depending on the polymer properties and its applications. These include nanospheres and nanocapsules, polymeric micelles, polymersomes, dendrimers, as well as polymer drug conjugates, to name a few. However, translating these nanomedicines to the clinic becomes a real challenge. A literature search on polymeric nanoparticles in Web of Science led to about 14,000 hits. However, the number in clinical trials and finally those that are approved is actually in double digits. Now, of course, there are a number of reasons associated due to which we see these nanomedicines not reaching the clinical stage. However, one of them is the difficulty in scaling these systems without changing their inherent properties. And we're going to see how microfluidics can address this issue. Now, microfluidics involves the mixing of fluids in the microliter scales. Consider two fluids which are flowing in a laminar condition, that is side by side, as shown in the image on the left. Now, within this laminar flow, one can further embed structures within the wall of the channel, and this will allow the fluids to fold over each other. This folding over exponentially increases the interface between these fluids while decreasing the diffusion distance, and as a result, the mixing speed becomes extremely fast. The nanoparticles that are formed here would be based on the principle of nanoprecipitation. You put your aqueous phase, such as a water or a buffer, through one inlet, and your nanoparticle forming material, which is dissolved in a water-miscible organic solvent, such as ethanol, acetone, or acetonitrile, for example, through the second inlet. As the two fluids will mix rapidly, the change in the polarity of the solution causes the nanoparticle forming material to precipitate out, and as the mixing time is faster than the precipitation time here, it gives us the ability to generate smaller size nanoparticles. The process is reproducible since the same amount of the two fluids come in contact with each other during this mixing process, and parallelized mixers can be added, which enables seamless scale-up of the formulation. So during the formulation development stage, what are some of the points you would consider while preparing polymeric nanoparticles? The first would be to tune the nanoparticle characteristics such as size, and this would depend on the intended application of your polymeric nanocarrier. Second would be to test the encapsulation of your biomolecule of interest, its uptake, release, as well as its therapeutic effect both in vitro and in vivo. Third would be to be able to easily scale up your lead formulation which in many cases becomes difficult. Now, in order to further discuss how microfluidics can address some of these parameters during formulation development, we are going to focus on two widely used polymer systems. One is the hydrophobic copolymer PLGA, which is known to form nanospheres or nanocapsules. And the second is an amphiphilic block copolymer, which is MPEG PLGA, which usually forms micelles. Now, typically the procedure for forming polymeric nanoparticles using microfluidics would involve injecting the polymer dissolved in a water-miscible organic solvent such as acetone or acetonitrile through one inlet, while the second inlet is injected with the aqueous solution with or without a suitable surfactant. The two fluid streams would mix rapidly in the microfluidic channels, enabling the rapid and controlled nanoprecipitation of the polymer, which ultimately leads to the formation of polymer nanoparticles. Now, the microfluidic procedure here can be used to exploit certain process parameters and optimize your nanoparticle characteristics such as size. Listed below are a couple of process and formulation parameters that can influence your nanoparticle size as well as other characteristics such as encapsulation efficiency and so on. We'll focus on the ability to tune size. Now, looking at the process parameters, you see the total flow rate or TFR, which is a measure of how rapid the mixing process is. Higher the TFR, faster would be the mixing. The aqueous to organic flow rate ratio, or FRR, dictates the number of parts of aqueous phase which would be interacting with the number of parts of the organic phase during the mixing process. So a flow rate ratio, or FRR, of 1:1 indicates one part of aqueous phase and one part of organic phase, whereas that of 2:1 indicates two parts of aqueous phase and one part of organic phase. The concentration of the polymer can usually affect the viscosity of the starting material, and the co-solvent approach is one where different solvents can be mixed to either aid in the solubility of your starting materials or aid in size tuning of your polymeric nanoparticles. There are a bunch of formulation parameters as well. Factors such as the polymer molecular weight, the modifications in the polymer chain itself, or changing the type of surfactant or the concentration of the surfactant can also influence the nanoparticle size as well. Let's see in the next few slides how changing these parameters influence both PLGA and PEG PLGA nanoparticles. So in the case of PLGA nanoparticles, you can see that the change in size on modifying the total flow rate or the aqueous to organic flow rate ratio, or FRR, is fairly minimal. As seen in the graph on the top left, increasing the TFR decreases the size from 150 to 120 nanometers, whereas on the graph at the top right, increasing the ratio of the aqueous phase shows an increase in the size from 130 to 160 nanometers. So these are fairly minimal changes in the size of the nanoparticles. However, on the other hand, if you look at the graph on the bottom left, increasing the concentration shows a high increase in the size of the nanoparticles, all the way from 70 nanometers right up to 200 nanometers. And this usually might be due to an increase in the viscosity of the polymer, resulting in a slower mixing, which is leading to larger size nanoparticles. Now let's look at PEG PLGA nanoparticles. So in this case, you clearly see that the total flow rate has a great impact on the size of the nanoparticles, as seen on the graph in the top left. Here you can see that increasing the TFR decreases the size from 80 to 40 nanometers. The aqueous to organic flow rate ratio, on the other hand, which you see on the top right, has no influence on the nanoparticle size as seen in the graph. Concentration, unlike the PLGA, has no impact on the nanoparticle size between 2.5 to 60 milligrams per ml, as seen in the graph on the bottom left. However, it increases significantly as the concentration goes greater than 60 milligrams per ml. Hence, what you're seeing here is that changing these process parameters will influence different polymeric nanoparticles such as PLGA and PEG PLGA in a different manner. Like for example, total flow rate did not have a very drastic change in the case of PLGA, but it's having a great impact in the case of PEG PLGA. The co-solvent approach, which you see on the bottom right, gives you an added flexibility to change the size of your nanoparticles without changing the total flow rate. This could be to either increase the solubility of your drug or polymer of interest, or it could be just to speed up the manufacturing process. For example, if you look in the graphs and you see formulations A and B, so both formulations A and B will give a similar size of about close to 80 nanometers. However, formulation A, which is in 100% acetone, is run at 1 ml per minute, whereas formulation B, where you're using a 25% acetone and a 75% DMSO based mixture, is run at 8 ml per minute. Now, at the lab scale, this would be fine, but as you scale up your manufacturing, the formulation B will be 8 times faster than formulation A, and this loss in time during large-scale manufacturing can greatly impact the cost. Of course, care has to be taken that using co-solvents does not impact other characteristics of your formulation. Now let's look at the strategy to maintain size after formulation changes. So having a look at some of the formulation parameters, in the case of PLGA nanoparticles, one of the reasons to change the characteristics of your polymer would be to influence the final delivery of your drug. For example, increasing the molecular weight of your polymer is expected to slow down the release of the drug, whereas changing the lactic acid to glycolic acid ratios can change the degradation profile of the PLGA nanoparticles. However, this change in the polymer can also change the nanoparticle size, like in the example shown here from the literature, where changing the terminating group or the molecular weight increase the size of the nanoparticles from 100 to 155 nanometers. Now, the size can greatly influence its pharmacokinetics through the body, and hence changes in the polymer characteristics, although desirable, might not be possible due to increased size of the nanoparticles. However, when we use microfluidics for a similar study, we saw that the molecular weight did not seem to influence the size of the nanoparticles, as can be seen here between a PLGA of molecular weight 100 kilodaltons and a PLGA of molecular weight of 12 kilodaltons, as compared to the graph on the left, where you see an increase from about 100 nanometers for a polymer which has a molecular weight of about 12 kilodaltons to about 155 nanometers for a similar polymer, but which has a molecular weight of about 45 kilodaltons. Similarly, changes in the terminating group, as seen on the left, or the lactic acid to glycolic acid ratio, as seen on the graph on the right, did not seem to influence the nanoparticle size, allowing one to make changes in the polymer and at the same time control the size of the system. Similarly, in the case of PEG PLGA, numerous reports in literature have shown that an increase in the molecular weight of the hydrophobic block in these amphiphilic block copolymers such as PEG PLGA or PEG PLA or even PEG polycaprolactone leads to an increase in the size of the nanoparticles. This is similar to what you see in the graph at the top. So you can see here that using a PEG of about 5,000 molecular weight and changing the molecular weight of the PLGA block going from 10,000 all the way to 95,000 increases the size of the nanoparticles from about 30 nanometers to about 60 nanometers. Again, increasing the PLGA block in this case tends to increase the stability, reduce the critical micellar concentration, and can also affect the encapsulation and release of the drug. However, it may not be always desirable if the size of the nanoparticle changes like it's happening in this case. So here again, using microfluidics, we can have control on the process parameters, and you can modify the total flow rates of the system as seen here. in the bottom graph, and you'll be able to generate nanoparticles of similar sizes, which in this case was 60 nanometers. So just by modifying the total flow rate, going from 0.75 ml per minute all the way to 8 ml per minute, you can actually get all the nanoparticles to a similar size of about 60 nanometers. Next, let's take a look at encapsulation of drugs into polymer nanoparticles. Now, polymers can be used to encapsulate a number of molecules such as hydrophobic and hydrophilic small molecules, proteins, peptides, and even nucleic acids. But for the purpose of this webinar, we'll focus on two hydrophobic drugs, which are coumarin 6 and vertiporfin. So coumarin 6 has a log P value of about 5.2 with a molecular weight about 350, and vertiporfin has a log P of 2.1 with a molecular weight of about 719. Now, this slide shows the encapsulation of coumarin 6 in the left graph and of vertiporfin in the right graph in PLGA nanoparticles and at different flow rates. Here you see that different drugs will encapsulate to different extents in PLGA nanoparticles. For example, coumarin 6 showed a drug loading of about 0.18% weight by weight, which is fairly low. However, if you compare that to vertiporfin, it showed a drug loading of 6% weight by weight. Hence, depending on your drug and your polymer system, different drugs tend to encapsulate to different extents. Another thing, however, to note here is the encapsulation is both these encapsulations are comparable to what we found in the literature. However, the size in this case is about 80 nanometers, while that in literature is about 160 nanometers. In the case of PEG-PLGA nanoparticles, we compared the encapsulation of coumarin 6 into these nanoparticles using either a co-solvent evaporation approach, which is widely seen in the literature, or we used a microfluidics-based system. And what you can see here in the graph on the left is that the size using microfluidics is much smaller at about 40 nanometers compared to 60 nanometers for the co-solvent evaporation approach. However, when you look at the encapsulation, the microfluidics method showed a significantly higher encapsulation at 60% compared to the co-solvent evaporation method, which showed an encapsulation efficiency of close to about 40%. This increase in encapsulation using microfluidics has also been seen when we've tried encapsulating coumarin 6 in liposomes as compared to what it was in the literature. And it's also been reported it's also been reported in the literature by other users who've used microfluidics and have noted higher encapsulation of other small molecules, for example, propofol in their liposomes when compared to some of the other conventional methods used outside. Next, we'll talk about the scalability of these nanoparticle formulations. We'll discuss using two instruments. The first, to your left, is the NanoSystems benchtop, which is used in early development and can make volumes about 1 to 15 ml. So this would be used during your investigational phase when you're trying to investigate the size parameters as well as the encapsulation of drug and doing initial in vitro in vivo studies. The second is the NanoSystems Blaze, which is used in preclinical development, and it can make volumes of about 10 to 1000 ml. Now, think of a scenario where you make a 2 ml formulation to tune your particle characteristics and investigate in vitro studies and some preliminary in vivo work. And now to do a slightly larger in vivo study, you make larger volume batches of about 10 ml formulation volumes. Now, this is in the case of PEG-PLGA nanoparticles. We used a co-solvent evaporation approach and found that just changing the volume from a 2 ml formulation to a 10 ml formulation led to a change in the size of the nanoparticles from about 93 to about 78 nanometers. This change in size can definitely influence your nanoparticle delivery. However, when using microfluidics, you can see that there is no change in the size of these nanoparticles. Now, how about scaling that even further? So in this case, we are working with PLGA nanoparticles. And if you look at the graph on the left, we made a 2 ml formulation of the PLGA nanoparticles using the NanoSimbler benchtop, and then scaled that up to a 280 ml formulation using the NanoSimbler blaze. And what you see here is that there's no change in the size and the PDI of the nanoparticles. Then we took the same nanoparticles at a 1x concentration that you can see on a graph to the right, and concentrated it using ultrafiltration to a 10x concentration, and you can see that there's only a marginal increase in the size and PDI, indicating the scalability and that both the blaze and the benchtop give similar nanoparticle characteristics, even though they are formed at different volumes. So in summary, the microfluidics enables you to tune your nanoparticle characteristics such as size depending on your intended application. You can also change the formulation parameters and still end up controlling the size. It can successfully encapsulate hydrophobic small molecules into your polymeric nanoparticles, as well as you can seamlessly scale up your lead nanoparticle candidates.
Thank you, Dr. Garg, for this excellent presentation. We do have time for a couple of questions from the audience. The first one here, how do you remove some of the organic solvent that is encapsulated in the nanoparticles during preparation?
So the removal of solvents would be done via appropriate post-processing, such as dialysis, or the other option is to use centrifugal filtration. Now, the sizes which you saw in the webinar were all post-dialysis. We did not see any significant change in the size of the particles when we tested them post their microfluidics as well as after doing the dialysis. The only difference we saw was that we saw a little bit of swelling. We saw a few. We saw a slight change. We saw a very minimal change in the size, and we think this could be due to the swelling of the particles in the organic solvent, and then there's slight shrinking on removal of the organic solvent. For like larger scale batches where you make like the 280 ml formulation, or if you go to like a liter scale formulation, tangential flow filtration or TFF is an option that can be used. I believe the previous two webinars from our company had a lot of information on the TFF, and I believe these should be up on our YouTube channel, so if anyone's interested, they can look it up from there.
Thank you. Second question here. Is the microfluidic system only good for hydrophobic drugs?
So I understand that what we showed here was mainly for using hydrophobic drugs, but no, the microfluidic platform can be used for a number of other molecules and not just hydrophobic drugs or hydrophobic small molecules. We have customers who successfully encapsulated nucleic acids as well as peptides, and currently in-house we're investigating encapsulation of proteins into polymer-based systems. Then that's just for the polymer side, but on other platforms, for example, on the lipid-based systems, we have a lot of customers and we have a lot of in-house data on nucleic acid encapsulation and delivery of lipid nanoparticles. But we've also had customers who successfully encapsulated both hydrophobic and hydrophilic small molecules into these liposomes as well. So yeah, it's not just hydrophobic drugs. We do have data for other drugs and biomolecules as well.
And I believe that's all the time we have for questions. Thank you so much for joining us today. If we didn't get to your questions, we will answer them via email after the event. If you have any questions or would like to learn more about the NanoAssembler platform, please visit www.precisionnanosystems.com. And don't forget to follow us on LinkedIn and Twitter. From Dr. Garg and myself, have a wonderful rest of your day, and thank you for your time. Cheers.